So basically, the talk is based on a paper we just published in the Journal of Clinical Oncology, Cancer Informatics. It was a team effort, so I am one among many other individuals who were involved in this team effort. Let's just literally, very briefly about all of us. We are a part of precision medicine. We are in NIH at the Office of the Director. And the idea is to try to collect close to 1 million EHR records on individuals, including information about physical activities, including information about genomics. And we are almost there. And basically to make it available for public through an agreement with institution to use the data. The study objectives were developing and operationalizing an electronic health record data quality framework. Although there are a lot of work done, we try to look at it in terms of all of us data, and to apply the dimension of this framework to ductal carcinoma in situ. And lastly, we created a checklist to guide us if we indeed reached our goal. Why is it significant? Because it provides information about fitness of use of our EHR data, and corollary, understanding the strengths and weakness if you use a checklist to make sure that you captured all the data that you are interested in. So why did we select ductal carcinoma in situ? Because number one, it's a pre-cancerous condition. It affects around 25% of females in this country. It's a well-known, understood disease and shows excellent response to therapy. And lastly, the incidence of ductal carcinoma has increased because of the use of more frequent use and an early age use of screening mammography. So what was the study design here? We tried to go in a little bit different way, and we actually created a case. And the case was basically earliest occurrence of any ICD-9 and ICD-10 or SNOMED, concept for ductal carcinoma, and map those concepts to OMAP. And the individual had to be female assigned at birth, as well as 18 years and older. I do realize that some males get the same phenotype, but we focused on females. And so this was the case. The control were individuals, the same age group, but without the phenotype. And for clinical measurement, we followed NCCN, which is an internationally known guidelines how to treat and how to diagnose. And it is very clear in this disease because it is less dependent on TNM, which means tumor, node, and metastases, because it's basically zero for node and metastases. And the data quality is, we selected, it's kind of a hybrid of many other. We had quite an extensive committees meeting trying to establish the data quality. And we came up with conformance, completeness, concordance, plausibility, and temporality. And again, I can answer questions, and please read the article because much more information is available there. And just some very brief definitions of the above is conformance, basically data values and elements, the standard, the syntax, and structure were equivalent or represented in the same way. And completeness, do we have enough data values to make decision, to analyze the data? Concordance, is the data value and elements similar or in agreement, okay? And we'll get back to it because I will make it very clear with a real example later on. Plausibility, does the data make sense? And temporality, is a temporality to the data. People are diagnosed, have a biopsy, diagnosed, get treatment, etc. Before we go on, and again, I will give you a good example of it, but we used a data quality dimension. The data quality dimension are conformance, completeness, concordance, plausibility, and temporality. And we looked at it along three dimensions. Concept selection, did we selected the right concept to assess fitness for use? Is there internal verification? Does other parts of the data in the EHR actually verified we selected the right? And is there an external validation to the data? And it is a yes or no. There is no score here. And hopefully in the future, if we get enough information of other phenotypes, we'll be able to start creating a scoring system. A little bit about the demographic and geographical distribution. We basically, our case were 2,209 females. The majority of them, 52.5%, are ages 60 to 79, which is appropriate for the biology of this cancer. And 66% were, guys, I will try to respond to comments as they go at the end, okay? And again, 66% were white females. And what's another importance here is that we don't have information about this specific phenotype across the nation because we are depending on academic institutions and other participants in our studies. So as you can see, 70% came from California and 12% from Massachusetts and Pennsylvania and Illinois, which means if you're looking at it from a national perspective, it's not, you won't be able to generalize. In terms of conformance, assessing the sources of distribution, the OMAP concept, so ICD-9 and 10 only represented 87% of the entire OMAP. And ICD-9 and 10 and SNOMED represent another 277 mapped to OMAP. And SNOMED were only less or equal to 20 mapped to OMAP. Completeness, again, just to remind you, completeness, do we have enough information? And what we did here, and it's very clear, the case has the information, the control has very little information. And for example, biopsy, 53.5% have biopsies in the case and only 3.2% in the control, which makes absolute sense in terms of looking at cancers that need a biopsy in order to establish the diagnosis. And this is an example of external correlation, so external validation. So let's go back on it. We selected age because I showed you before that age is very pertinent, very important biological marker or risk factor for this cancer. And then we went, so the selection was done, let's look at age. because we know age from internal verification is the right parameter to use, then go ahead and look at external validation that indeed we selected the right concept. And as you can see, it's very striking that the correlation is very high. And indeed, in our case, we did select the right appropriate parameter. Plausibility, remember, does it make sense? So we went ahead and looked at case and control. Again, remember, age is an important marker. So we looked at clinical measures based on NCCN, clinical measures and intervention based on NCCN, and we compared the case to the control, and the picture actually speaks for itself, that indeed it seems that we selected the right cohort, because it's quite statistically significantly different and visual difference between the case and control. Temporality, and temporality is the idea that information has some sequence to it and has some logic to it. So I can tell you the antecedent of biopsy to data which were available for around 47%, and the median time to biopsy to diagnosis was around 8 weeks. The mean was, it was a very skewed distribution, therefore we selected the median. And remember, we talked about NCCN phases, and indeed for most cases, it followed the NCCN phase, okay? And which means workup was mostly prevalent. And again, I'm sorry if I don't know exactly how to describe the color. It's orangey type, actually represents the workup. And the green represent the treatment, which is surgical treatment. And post-surgical treatment is in blue. So we didn't have absolute complete right sequence, but overall, we had the right picture that follows the NCCN guidelines for clinical measurement and intervention. So what is the key finding? What is the take home for me? We did develop a systematic generalizing approach to data quality, and we were able to implement it on a case, on the test case, which was very contained and known, basically. All 5 dimensions were evaluated. Unfortunately, we didn't have a lot of external validations, and it did, in one case, as I showed you with concordance with age, did have an external validation. And we use case and control, which is something that is very valuable and very unique in terms of our work, in terms of our data, rather, to see if indeed we selected the right indicators, the right clinical measurements and intervention, and indeed we had the right case in front of us to assess fitness of use. Okay, limitation, we are overrepresented by academia. Academic institution, there is a fragmentation because an individual may get care in one institution and may get follow-up care in another institution. And we don't really have data from notes, which will be, or biopsy results, which will be very important if we went ahead and looked at other cancers. And next step is really the application of the framework to other phenotypes, and we are busy doing it. And it still works very well. And just refinement of the temporal analysis, because we need to figure out to show that indeed there is a progression which is logical from biopsy to cancer diagnosis to treatment if you use diabetes or hypertension or any other disease or any other phenotype. Basically, it is the same type of issue to show progression. And this is me, and feel free. They didn't put the RN. I'm a registered nurse in addition to being a PhD. So again, feel free to communicate with me, and I will share those slides later on. I will send it to the organizer so you can look at it. Thank you. Thank you very much.
Thank you